Hello, my name is David McDonald. I'm going to present my research on wavelength effects on UV disinfection and photorepair of E. coli in solution that I've done in Dr. Natalie Hall's lab. UV radiation can be used to disinfect water. This can limit the need for chemical disinfectants like chlorine. One mechanism for UV disinfection is through DNA damage, which occurs when cyclobutane pyrimidine dimers, or CPDs, form between two pyrimidine nucleotides in a DNA sequence, as you can see in Figure 1. The low-pressure mercury lamp is the standard for UV disinfection. Figure 2 shows how the peak emission of the low-pressure lamp at 254 nanometers overlaps with a peak of DNA absorbance. Protein absorbance is much higher at shorter wavelengths. The krypton chloride Xe-lamp emits light in this range, with a peak wavelength of 222 nanometers. The krypton chloride lamp is also mercury-free. Photorepair is the ability in some bacteria to undo UV-induced DNA damage in the presence of light. E. coli is one organism with DNA photolyase enzymes, which repair CPDs when exposed to light with wavelengths between 300 and 500 nanometers. The absorbance spectrum of photolyase, shown in Figure 3, overlaps with the output of common light sources like fluorescent lamps. This research seeks to find if less photorepair occurs in E. coli after UV disinfection with the krypton chloride lamp compared to the low pressure lamp, because the lower wavelength light damages proteins like photolyase. Figure 4 shows the methods used to compare inactivation, protein damage, and DNA damage in E. coli samples exposed to UV light. E. coli were grown and suspended in a solution, then exposed to low pressure or krypton chloride lamplight. The amount of irradiance emitted by the lamps was measured by a radiometer. The UV fluence delivered to the sample is a product of irradiance and time, so the larger fluences were achieved by increasing exposure time. UV-exposed E. coli were cultured on agar plates, and the number of colonies were counted to determine cellular concentration. Concentrations were compared to a sample without UV exposure to calculate the log inactivation, as shown in the equation. Enzyme-linked immunosorbent assays, or ELISAs, use antibodies that bind specifically to the molecules being measured. Then a second antibody attaches with a conjugated enzyme that causes color to appear when a substrate molecule is present. ELISAs are being used to compare sample CPD concentrations. A photolyase ELISA is also being developed. The concept is shown in Figure 5. Absorbance readings measure the amount of color, which is related to concentration with the standard curve. Figure 6 shows two example standard curves from ELISAs. The results of 5 fluence response experiments are shown in Figure 7. The chart shows average log inactivation values at different fluences from multiple experiments. The arrow bars represent one standard deviation. The lamps produced similar results. Both achieved 4 log inactivation of E. coli with minimum fluences in the range of 12 to 20 millijoules per square centimeter. Results from each lamp had shouldering at low fluences and a tailing effect at high fluences. Tailing may be caused by cell aggregation that increases the bacteria's UV resistance at a high fluence. A CPD ELISA was run on samples from one of the fluence response experiments done with each lamp. Figure 8 shows the CPD DNA concentration from the samples, which received 40 millijoules per square centimeter of fluence. The results are reported as absolute CPD DNA concentration, CPD relative to DNA, and CPD relative to cell concentration as CFU. This is because the samples tested had similar but not equal amounts of DNA in cells. When reported as absolute concentration or as relative to DNA, the difference between labs is statistically significant, with the low pressure causing more CPDs. But when reported as relative to cellular concentration, the difference is not statistically significant. The low pressure lamp also caused more inactivation than the krypton chloride lamp in the experiments these samples are from. In conclusion, the krypton chloride lamp was found to produce similar disinfection to the low pressure lamp in terms of bacterial inactivation as measured by plate counting. The low pressure lamp did produce more CPD DNA than the krypton chloride based on CPD ELISA results from samples that received the same fluents. Though the low pressure lamp also caused more inactivation in these samples, and the differences in CPDs are not statistically significant when cellular concentration of the samples are accounted for. Future work on this project will include optimizing the photolyase ELISA to measure photolyase concentrations in UV exposed samples. This will be used to compare the protein damage done by each lamp. Photorepair experiments are being planned to find if the krypton chloride lamp limits E. coli's ability to repair CPDs and reactivate after UV exposure. Samples will be exposed to fluorescent lamplight after disinfection. The same analysis will be done to compare cell concentration, DNA damage, and protein damage. Figure 9 shows the krypton chloride lamp is hypothesized to cause less reactivation than the low pressure lamp. I would like to acknowledge all the help I received from PhD student Daniel Ma, who is doing similar research on photorepair but with UV LEDs. I was also able to do research this summer thanks to the CEGE Undergraduate Research Fellowship. Thank you and enjoy the rest of the research festival.